But those type of evidence are very, let's say, vague. It's not quantitatively related to its therapeutic effect. So in Gili, we have a comprehensive pharmacology database with all the experiment data. So we also did a database mining, which is a mining based on structured data, which is another type of technology. And all the work is done using Python. And this is an example of what type of data I was talking about. So for the drug discovery, normally we have in vitro and in vivo and clinical stage data, and on each stage they have different interpretation scope of how significant they are and how to interpret their quantitative result. For example, this is one of the SARS target, and its IC50 enzymatic acid values that and also each molecule has its smells, which is a representation for its molecular structure, so that we're very detailed on this type of chemical level information, so we would know which therapeutics works better than the other, and what are the potential issues or therapeutic effects of each medicine. So that we have dumped this type of data on our information sharing portal in late January, and I think a lot of researchers have looked at all this raw data and also helped us analyze from their perspective, and later have publication based on some of the data source. And we also did our own analysis and cleaning, so that we eventually focus on finding the to deeply analyze the viral targets, host targets, and all the therapeutics in a more thorough way based on different type of reasons. So this is gonna introduce the second workflow, that reasoning on known evidence. So the first rationale is species similarity, so that we know that SARS and MERS and the SARS-CoV-2 are in the same family, and they are very close. So, but we don't know, but a genetic sequence alignment similarity is not very specific. So we don't know which target is really similar. So that here I put out 3 targets that we built based on the protein sequence and using the Swiss Institute provided open source free tool Swiss model, and also on their server to build the initial homology model, because the sequence of SARS and the SARS-CoV-2 on those targets are very similar. So we would assume the homology model is relatively accurate. And some of the structure has been solved later by crystallographer and proved that the homology model, the error is very small. So that we can see from the 3 graph that the yellow highlight sequences are the ones that SARS and the new coronavirus are different. So that you will see for the C3-like protease almost identical, a tiny variation on the catalytic side. And for the RDRP, it's the catalytic side, which is right here, is completely conserved. But for pattern-like protease, there's a lot of difference on the 3' inhibitor side, so that we would see that the high priority is the RDRP target, and possibly the C3-like protease. And also later we know that RDRP is also the target of remdesivir, which is the most promising drug currently tested in clinical trial. So which means this approach, it does make sense. And then we also pulled out our database and cleaned 2 sets of data that is related to SARS and MERS. One is the phenotypic viral infection assay result. So those are cellular-based assays that have been tested on SARS or MERS, that those chemicals are effective against them, which we would reason that it has a higher possibility against the novel coronavirus as well. And this list is actually about 100, or over 100 molecules, and it's ranked by the most potent result from all the assays that have been tested. in the past. And also we find all the marketed RDRP inhibitors for various species as well. Hopefully they can be repurposed as the potential candidates. And some of the, I think some of the candidate was later tested in the real SARS-CoV-2 cellular-based assay was proved to show similar effect as they showed in SARS. And another thing is we want to find out the infectious path when we don't know anything about this new virus. We want to know how it's infect human. The only thing we can search is SARS, so that we use this website called the Gene Cards that they give if you search any pathogen's name, it will giving associate human gene and based on a score. The score is basically a measurement of how frequent these two concepts has been mentioned in literature, and then they give if they are associated more in the reports, that they give a higher score. So we know that for SARS, that ACE2 is the number one, and the score is way higher than the rest of the target. And we also know that TMPRSS2, the serum protease, is also a therapeutic target for some of for the SARS outbreak. So that which means this type of association algorithm, even without really reading any literature, can still give us a good hint of the key receptors that we need to focus on. And so that we quickly did another homology model based on the SARS structure. So for the SARS-CoV-2 spike protein and human ACE2, and also we perform a quick MD study and to see how they actually associate to each other. And the initial simulations that shows a very stable association, so that which this binding affinity is kind of correlated to the infection rate. And that's something we're very interested in and wanting to know at the early stage of pandemic prevention. But those are still very early stage of biophysics or simulation level experiment that it can really give any conclusive result, but it just gives some guidance. And later on this, the ACE2 was the binding affinity was measured and was proved to be really strong and hints to its high infection rate. So, and then the second rationale is the broad spectrum antiviral drugs. So the assumption is that if a drug can target different type of viral infection, no matter how different those two pathogens are, they may target similar host biologic pathways, or they may target on a viral target that the target itself is conserved across all species. So that we perform a data mining, and we digged about 7,000 antiviral compound if we're using like 1 micromolar EC50 as a cutoff as effective. And then we further identify 462 molecules has at least 2 virus species, and 51 has at least 5 virus species that they show efficacy. So this graph is showing basically this 51 molecules in orange, and how they inhibit different type of virus species. And this the detail of the data is reported on our information sharing portal. And we did a further analysis on all those drugs, and actually there are majority of them are indeed target human target, and which probably explains some of their broad spectrum activity. And also drug can be repurposed also because different target, even completely unrelated, maybe the drug binding pocket may share similarity as well. That's another reason of you see some of the drug can have multiple effects. So that based on that result, we get 2 lists. One is we get some of a relatively short list of known drugs, and then we get a giant list of early stage compound. And what are we going to do about the data? So we would recommend, for example, the shortlist contains dozen to 100 drugs that maybe experts can follow the literature deeper to dig and compare and maybe even make clinical recommendation. Because known drugs, his toxicity profile is already being tested, so that we would know how to use them in the correct therapeutic window. And also, but we need to make good use of the early stage data as well, and the data amount is really really big, so that we often would using those type of data to build AI predictive models, and in order to screening like new possibilities in a wider chemical space. So here goes to the next step, that we train and evaluate the AI model based on different type of set we collected, and each model is based on a particular scientific rationale.